I think screen is being, you can see the screen. Right. So when it comes to the basics of nucleus, discovered by Robert Brown, year 1831, and it's of course the life of the cell depends upon the nucleus. You can say the brain of the cell for that matter. It influences, controls almost every cellular activity that is taking place. So how is it shaped? Spherical or oval, and it has a like what do you call it? The vascular plants, RBCs, do not have nuclei upon maturity. That is an important point. And it is a membrane-bound cell organelle, and it controls the cell's growth and reproduction, almost all the cellular activities. And it is surrounded by a prominent structure which is known as the nuclear envelope. And this nuclear envelope is separating the nucleus from the rest of the cellular contents. That is the cytoplasm and the rest of the cell as such. So the cell's chromosomes, whatever are there, they're present within the nucleus. And this has its hereditary information, which is necessary for every what do you call it for every cell to divide and for the next generation to come up, along with every kind of activity to take place. So when it comes to the structure, it's around 6 micrometers and occupies nearly 10% of the cell volume, in fact. So that's quite an amount, in fact, if you consider. Of course, it's the endoplasmic reticulum, Golgi complex, and after that you have the the other cell organelles which are occupying space at the same time. This is occupying around 10%. So you see over here, this is the nuclear envelope, and you have the outer membrane, the inner membrane. So there's an intermembrane space also. And within the nucleus, you have the nucleoplasm, similar to the cytoplasm. And you have the nucleolus as well. And in the nucleolus, you have the chromatin network or the chromatin fibers. So anything that is taking place over here, the transport of substances from the nucleolus or the nucleoplasm is going to take place through the pores over here that you can see to the outside into the cytoplasm. And if you see the nuclear envelope, it is actually again double layered and encloses the nucleus, and at the same time it helps in passage of materials into and out of the nucleus. It's also known as the nuclear membrane, and it is in continuity with your endoplasmic reticulum. And you see over here that you have the outer membrane, the perinuclear space, the intermembrane. Of course, you have a pore. So this is going to be protein in nature. And this is known as your NPC or the nuclear pore complex. We will come to the structure of it in some time. And on top of the nuclear membrane also you have ribosomes, protein synthesis. All the proteins necessary for transcription, translation, all of them, most of them are being produced over here. The ribosomes is going to take place or participate in the protein synthesis. And nucleolus, within the nucleus you have nucleoplasm and the chromatin network and the nucleolus. So it occupies around 1/4 of the nucleus. What is it made up of? Again, proteins and ribonucleic acids. This is the sole reason that everywhere within our body, if you see, we are having a lot of proteins. And that is also one of the reasons why we need more amount of proteins also to maintain our health. And its main function is to again rewrite the ribosomal RNA, the nucleolus function, and then helps in protein synthesis. And this results in incomplete ribosomes being synthesized. Now we have chromosomes inside the nucleus, and these chromosomes, they are made up of protein and DNA. Now, in the nucleoplasm, this is something like your cytoplasm. So within the nucleus, it is known as nucleoplasm, and also you can name it as karyoplasm. So what is it made up of? Similar to cytoplasm, mostly water, different kinds of molecules, and ions. So it is again enclosed within the nuclear membrane. And this is one slide. This is the basics of it. So just like share, I will uh, share. Just give me data. Please give me a moment. Right, I'm going to resume the share. Just give me a There's so many slides that I am like total get confused. So that is what is happening. So please bear with me. Apologies. Right, got it. So share. Now I hope you can see the screen. Again, apologies for taking such a long time. Right. So when it comes to the structure of the nucleus, we've already seen it is like, in continuity with the endoplasmic reticulum, like I told. And at the same time, you have certain nuclear core complexes. In this, when you see, it is the largest cellular organelle in the animal cell, usually. And it's nearly 10% of the volume, like I told you, and is around 6 micrometers. And at the same time, it is irregular because of the presence of so much of DNA. Again, DNA is your acid, RNA, proteins. So almost 75% of the DNA, or 75 to 80% of the DNA, I mean of the nucleus is made up of proteins. Now, when it comes to that, we have certain nuclear envelope, like I told you, and then you have it consists of basically, yeah, nuclear laminae, and then you have the tubules and the inner and the outer nuclear membrane, which is continuous with the endoplasmic reticulum, and again phospholipid bilayer. And you have something called as a nuclear core complexes. I'm trying to put this down so that, right. So this nuclear lamina, fibrous in nature, proteins again, that is lamins, it is going to cover the complete nuclear envelope. So molecular weight is around 60 to 80 kilodaltons, and you have intermediate filament proteins, and you have three genes which are A, B, C lamin genes, which are going to encode your protein, which is going to give you the lamin proteins and the filaments. And then you have around two polypeptide chains, which are going to completely surround the nuclear lamina. And then this is going to bind to chromatin or DNA through histone proteins. I have a different PPT wherein I will share it as well so that you can have a look at it. Then this is going to be surrounding or forming the nuclear envelope and gives the mechanical support to the nucleus. At the same time, whenever there is DNA replication and cell division, then you will have the nuclear lamina actively participating in the processes. So there is one particular disease. We have already seen different movies. Probably there's one, just if I'm not wrong, there's this Hindi movie wherein Abhishek Bachchan, Amitabh Bachchan are there. So it's based on this particular syndrome, which is known as progeria. So in this, what is happening? LMNA genes, that is your laminate genes, that is lamin A, B, C, like I told you, there's a mutation, point mutation. So one amino acid that is cytosine is replaced with thymine, because of which just one particular amino acid is getting changed, mutated, because of which what is happening is there is prematurity or there is old age. And the kids will not live up to, or the children, whoever have this progeria, do not live up to a long life. So that is one thing about the nucleus. And when it comes to the nucleolus, it is the largest structure again, around one-fourth or 25% of the eukaryotic cell. Inside the nucleus, it is going to be occupied by the nucleolus. Not surrounded by membrane, has again DNA, RNA, different kinds of proteins, and it is the site of ribosomes synthesis. And it has the genes encoding your 5s, 18s, and 28s ribosomal RNA. So anything related to ribosomal synthesis is taking place in the nucleolus. And when it comes to the nuclear pore complex, you can actually see it is made up of different kinds of proteins. So this is the cytosolic phase or the outer membrane of the nucleus. This is the inner membrane of the nucleus. This is where you have the nuclear pore complex. This is the nuclear envelope. This is the luminal or the intermembrane space present inside the nucleus, cytosolic end, nuclear end. So what is happening is over here you have a ring-like structure and filaments which are facing towards the cytosol, and over here you have a pore ring. Please imagine this is the transverse section, so you can actually see the half that is this. It is a cylindrical tubule kind of structure, and this is the central transporter, and this is the pore ring, and you have a nuclear ring this side and a cytosolic ring and luminal ring. So there are three ring-like structures over here if you notice, and this is the pore ring which is present inside surrounding the central transporter. Now, all of this are known as the nucleoporids, and this is through which all the molecules, ions, macromolecules can travel between nucleus and the cytoplasm. So, this is the basic structure of nucleus, nuclear envelope. Now, I will talk about, this will come later. Structure and organization of chromatid chromosomes. We are done with. We'll come to nucleolus later. Molecular organization of chromosomes also is going to be there. And nucleosomes packaging is going to be important because, for the packaging of the DNA and why is it necessary, we will just have a look at it. And I'm not going to go in order. You please try to understand that, whatever, is being taught in your textbook. So we will just have a look at it. So when it comes to the structure and organization of chromatin, we have different models that are going to give what is chromatin. Now, how is it important to us? Now, we know that nucleus is necessary for the functioning of the cell. So, what is chromatin now? It is the combination of DNA and proteins that is going to make up our chromosomes. And how is it visible? By staining. Certain staining techniques are there. So, that will help us look at them and know that yes, this is chromatin. So what are the major proteins that are going to make up the chromatin? They are the histone proteins. And what is the function of the chromatin? It is to help package the DNA into a smaller volume so that it fits into the cell and also gives the DNA the strength to allow mitosis and meiosis to take place. So that is, and also it helps in controlling the gene expression and the DNA replication. So what does it have? What is the ultrastructure of chromatin? If you see, then through the electron microscope, we still have not got a good or a clear picture of organization of DNA in the chromatin. So we have a few hypotheses that have come up to know how the packaging of DNA in the chromatin has taken place. Now, studies have been, have shown that it's around 2 nanometers thick and 2 nanometers in wide, single fibrin consisting of a single DNA molecule. Now, this is the nuclear pore complex. This is the chromatin network that is present inside the nucleoplasm. And then this has a structure that is something similar to a solenoid, which is a magnet. So, loop-like structure consisting of nucleosomes, the DNA helix, and the histones over here. And you have a centromere, and telomeres are the ends of the centromere and the condensed chromosome. So different models have been proposed to describe the organization of the DNA in the chromosomes. So when it comes to that, chromatin fiber is around 100 angstroms in diameter, and two strands are present with a diameter of around 35 to 40 angstroms each. Now, it consists of a double helical structure, and four chromatin fibers are wound towards each other to form a quarter chromatid. So, and then this quarter chromatid is the smallest subunit of the chromosome and it is around 400 angstrom. So, this is what is given in the multi-stranded model. So, to form all of this, chromosomes finally have around 64 DNA double helices. So, that makes around 3200 angstroms in diameter. So this is one multi-stranded model. In the folded fiber model, folded fiber model, this was proposed in 1965. Again, chromosome is tightly folded, having around 200 to 300 angstroms in diameter. And it is coated with histones and non-histone proteins. And folding of the chromatin fibers will reduce the length of the chromosome. So this is by far the most popular model that was proposed. Now in 1974, Kornberg had proposed the nucleosome model. According to this model, what he said was the DNA, whatever is present, is tightly bound by histone proteins and in a repeating manner. And this DNA protein is called as the nucleosome. And this is by far the most widely accepted model. And let us just see how this organization of the chromatin is and how it is packaged around the DNA. So it's like a bead on a string structure. 30 nanometers condensation is taking place. And when it comes to DNA, what you call mitosis or meiosis, you have a phase called metaphase in which there is a much more compact and a higher degree of packaging of the same nucleosome. So it's in a threefold manner. If you see, finally you have the cell. This is the cytoplasm, nucleoplasm with around 46 chromosomes. So each chromosome is going to be a tightly packed with what? DNA. Chromatin DNA. DNA is packaged or packed with the histone proteins. This forms the double helix. And this is how genes are present on the DNA. This is the structural organization of chromatin. So when it comes to nucleosome, what is it happening? In eukaryotes, DNA is bound by the nucleosomes. Histones are going to pack the DNA in a very orderly manner. And this fundamental packing unit gives the chromatin a bead on a string like structure, and this is how you are going to see in a electron micrograph. And how are they linked to each other? By a linker DNA. So now we have 5 types of histones called as H1, H2A, H2B, H3, H4. The histones are the basic proteins that are present usually. So you have why and one more thing is basic in nature. So their amino acids are around 24%. That is one-fourth of them have lysine and arginine. They're not acidic in nature. They are basic. That is what they mean by basic. So they're not simple as you feel, but it is the basic, acidic, basic. So histones have positively charged amino acid chains, and this is bound to the negatively charged phosphate group of the DNA. So there is a core protein and that is having 8 histone molecules in fact can be positively charged with lysine, positively charged lysine and arginine molecules also. And so you get to see this is the DNA, this is the core protein, you have the linker DNA over here. So this is the histone H1 over here. So this forms the nucleosome. So whenever you use an enzyme that can degrade DNA, any kind of deoxyribonuclease or enzyme micrococcal nuclease, any of these enzymes, then the DNA string can be digested. So only the linker DNA is getting digested over here or degraded. Rest of it is protected and it is going to remain as a double-stranded DNA and around 146 base pairs fragments bound to these 8 nucleosome histomers. So it forms an octamer is going to remain as such. Now each nucleosome, this is known as a nucleosome. So each nucleosome has a disc-shaped particle, and it has a diameter of around 11 nanometers and around 5.7 nanometers in length. And core proteins, see these are the core proteins. Around 8 core proteins are present. So this proteins, it has two discs arranged in parallel, four on top, four on the bottom. Now four core proteins are H2A, H2B, H3 and H4. So you have four protein molecules on each disc. So eight in total. So this DNA molecule runs along the disc. Please imagine it in such a manner that you have two halves of an orange, for example, and then you cut it, and then the DNA, and then you once you cut the two halves of the orange, you're going to make that the top half, whatever is there, cut into four more pieces. Transverse section, and then horizontal, and the longitudinal section, two longitudinal cuts. So, and then imagine them in this manner, and to tightly bind them, you're going to use DNA. So this is how you have to imagine over here. So this is one half of the disc, second half of the disc wound by the DNA. This is the linker DNA again. And this is the histone H1 over here. This has H2A, H2B, H3 and H4 molecules over here. So they make two. One H2A, two H2A in this manner. Now, once this done, the DNA molecule runs along the rim, acts as a seal, and around 146 base pairs of DNA are associated with this nucleosome core basically. So in total, you have around the length of the DNA also, which is around 60 base pairs. So totally, if you see 146, 60, in total you have around 200 base pairs of DNA. And DNA is going to take two complete turns around that histone octamer, and two turns are sealed by the H1 molecule as well. They are going to actually be sealed. How are you going to stop the DNA molecule from unwinding? This is done by the H1 molecule. The seal will be remained with the help of the H1 molecule which is binding it. H1 acts as your cellulose. So around the nucleosome, it's going to repeat at intervals of 200 nucleotides or base pairs, and this is how your basic packaging is taking place. These are your histone proteins, octamer, and this is your linker DNA that is present over here, and this is the DNA wound, and this is your histone 1, and this length is around 55 angstroms, and this is around 110, and from here onwards, this is the linker DNA which will get degraded with the help of nuclease. So this is one thing. I do not have a picture over here. Anyway, you have around 147 base pairs of DNA wrapped around the nucleosome. When it comes to the chromatin fiber, this is going to be lysed by the any kind of nucleases, and diameter is larger than that of the single nucleosome, suggests that the nucleosomes are packed or organized in a very compact manner. And 30 nanometers of this fiber, actually chromatin fiber, is closely packed nucleosomes. This is going to be in a solenoid structure, that is around 6 nucleosomes per turn. So you have to imagine this, and in this manner you have a solenoid, that is magnet type of structure. Unfortunately, I do not have that picture over here right now, but otherwise in a ring like manner, and many rings together, and this fiber formed by the H1 that is going to be bound by the linker DNA enters and leaves the nucleosome. So finally, you have that solenoid structure also forming the chromatin network. Now, binding of the H1 molecule to the chromatin leads to a creation of a polarity. So you have charged polarity that usually the normal chromatin doesn't have. So this is the histone core. Around 6 of them are wound. This is how it is going to be. This is the what do you call it, solenoid structure. So how are you going to imagine? We have DNA, and these are the nucleosomes, octamer, that is going to be present, surrounded by the DNA. Then the histone H1 is going to be there, which is going to help in attachment of both the DNA coils. You have a linker DNA, and plus you have a spacer DNA over here along with his H1 histone. And this is going to be in a wound structure. Around six histone molecules or six nucleosomes are going to be present in a compact manner, having a looped domain, which are again linked over here with different kinds of bonds, in fact, based on the polarity that is present. And this consists of your chromatin fiber. So when you decode or remove or unentangle your chromatin fiber, this is how you're going to get. So the final structure is going to be something of this sort, which if you simplify it, and this when you supercondense it, you're going to get the chromatin fiber, which is around 300 nanometers in diameter. This forms the chromatids. This forms the centromere, and then your arms in the chromosomes. So this is around 1400 in nanometer diameter, in fact. So shortly region of the DNA double helix is around 2 nanometers. And on the string, this particular part, if you see, it is the linker DNA. And these are the octamers of the histone proteins, in fact, surrounded by DNA. This is around 11 nanometers. And when it is packed in the form of a nucleosome, this is around 30 nanometers. And this is how you're going to get it in the chromosome in an extended form. And when it is condensed, it is going to be in this form, 700 nanometers. And the entire mitotic chromosome is going to be in this manner. Completely condensed 1400 nanometers. When you actually see how it is going to be, this how it is going to be this one. So it is an interesting way of how the DNA is packaged, and all of it is present inside the cell, inside the nucleus, inside the nucleus. So when it comes to the metaphase, wherein we have DNA division, right, cell division, we have to have a much more condensed manner. So increasing the packaging is increased packaged DNA is observed in the metaphase chromosome. Especially it is the highest in the metaphase. So to prove how it is done, they have performed an experiment wherein the histones from the metaphase chromosome were removed by adding a polyanion dextrin sulfate. Then they saw that the histone depleted chromosomes that were there have a central core scaffold surrounded by a hollow mode of loops of DNA made up of non-histone proteins, and still the general shape of the metaphase chromosome was being maintained. Now, when the histones were removed, DNA that was packed became extended, and you could see that the average length of it was around 25 micrometers, and it had around 15,000 base pairs. So based on this, they actually could make a model of the chromosome structure also. This was done in the early 1900s to late 1900s, around 1979, in fact. And they did further experiment, and then they one more what do you call it? What they did was they proposed a loop model in which the DNA is anchored or arranged in the loops. This is attached to non-histo or scaffold. Now, when they did that, what happened was the lateral loops were found to have 25 micrometers in DNA, and it was contracted 40 times, and it was present in a 30 nanometer fiber, which is around 0.6 micrometers long. So this length was consistent with the metaphase chromosome, which is around 1 micrometers long. So, in this manner, they understood that in the early stages of cell division, chromatin strands are going to get condensed, and then they are not going to act or help in DNA synthesis. That is, transcription is going to be stopped, and then it is going to get more condensed and visible. You have the forearm structure that is going to be present, and we are talking about over here uh, the division of the cell as such. So you're going to see the chromatid, sister chromatids, and the central centromere that is going to be there, and the cell is getting ready for cell division. And you can see other changes such as the microtubules coming, attaching to the centromere in the center so that the whole cell can be pulled from the centromere to the opposite sides of the cell. Now the microtubules, once they do that, every daughter cell is going to inherit one set, short arm, long arm, plus half the chromatid centromere. So because of which you have the inheritance of the genetic material. Once this is done, again the chromatids are going to uncoil themselves, and then they are going to function as chromatin network, which is completely inside the cell. So in spite of this, basically all the chromosomes are going to be highly condensed, and this giant structures of the DNA is going to be visible inside the cell. So this is the DNA double helix. You can see the histone, nucleosomes arranged in a solenoid manner, and this is going to be the chromatin loop. All this is actually how it is going to be present in this particular chromosome. So now we have two types of chromatin over here. What is it? Euchromatin and heterochromatin. On what basis are we calling such kind of chromatin, or on what basis are we distinguishing them? Based on the method or way they are going to get stained. So euchromatin is rich in gene concentration. It's going to take a lighter stain. So you're going to see it in a lighter manner. And once mitosis is finished, you cannot see euchromatin anymore. So what is euchromatin made up of? So structural genes. So genes that encode for the structural framework, and it is the genetically active chromatin. In fact, it's around 3 to 8 millimeters fiber. I'm talking about millimeters now. And during metaphase, it takes up the darker state because it is going to become the chromatid. So if it is during non-replication time, then it is going to be euchromatin, going to be lightly stained in fact, and it has the structural genes. Heterochromatin, this same chromatin, which is euchromatin, when it is during the cell division, it is going to be tightly packed, takes in a darker stain. So you get a deep color during the interphase and prophase. But during metaphase again, it's going to get lighter. So chromomeres, centromeres, and nerves look darker. They because of the condensation and tightly packaged way they are, and they are the true heterochromatic chromatin. So over here again, chromomeres, whatever are present, are not the true heterochromatin in fact. In the chromosomes, all the centromeres that are present are going to form this heterochromatic mass known as the chromocenter. And again, it is highly repetitive in nature, and you cannot see this during the late S phase stage. Again, heterochromatin is of two types: constitutive heterochromatin, facultative heterochromatin. What is happening? Heterochromatin, in the constitutive heterochromatin, it is permanently inactive. It is going to be in a condensed state throughout the cell cycle. We know that cell cycle has a G1, S, and G2 phase, and of course you have a G0 phase as well. Anyway, throughout the cell cycle, it is going to be inactive. And most common type of this heterochromatin is near the centromere, in the telomeres and in the C bands of the chromosome. So they are going to be darker state. So the telomeres are the extreme ends of the sister chromatids that are there, both the short arm and the long arm. You have C bands that are present. So they all take up a darker state. Now, constitutive heterochromatin is having short repeated sequences of DNA known as the satellite DNA. This is because it is called as a satellite DNA, as once you do the ultracentrifugation, you can see that the same DNA is going to get repeated. So, and this forms the main. Again, you have different kinds of DNA over here. Satellite DNA is one, and then, depending upon the fragments that you are going to get, so you can do like, RFLP analysis, RAPD analysis. Maybe we'll come to that in some time, or you must have already learned it, at least the basics of it. So I'm not going to go into it. When it comes to facultative heterochromatin, then heterochromatinization is what the process is that the DNA, the heterochromatin or the thicker chromatin has undergone. So again, it is in a permanent condensed state, and again it gets dispersed whenever there is transcription takes place. So one pair of chromosome becomes partially or totally heterochromatic. Example is X chromosome if it gets inactivated in female mammals. So what is happening is this is gene silencing or chromosome silencing that is happening. And that X chromosome, if it gets silenced, other is packaged in the euchromatin and it gets expressed. So the silent chromosome, whatever is there, is inactive, forms the interface sex chromatin or the bar body. It was named after the so you have different kinds of this bar body is not going to be transcribed. So since only one X chromosome is there and it had got silenced, you cannot find this in the males. So when it comes to the composition of chromatin, made up of DNA, made up of proteins and RNA, because you need for RNA synthesis also, right? And the composition varies according to different species as well. And whatever DNA is present in the chromatin is going to be the DNA that is going to control the heredity. And you are going to measure the DNA amount in picograms. Nanograms 10 to the power of 9. Pico is 10 power minus 12 if I'm not wrong. Now we have something called as the C value. Again, I have another presentation, in fact, which I want to share with you. The C value paradox and all of that. So when it sounds very, very nice, in fact, the C value paradox. So the basic definition of paradox is unnecessary event, or like, what do you call it? Uh, I'll just have to tell you just a second. So it is a seemingly contradictory statement over here. So this is about the C value actually tells you the amount of DNA content that is present in the diploid organism. So if at all, organism that like, whatever the DNA that was stained and the amount of DNA in a single nucleus was measured using a cytophotometer. Now, this showed that a constant amount of DNA was present in different organisms. So, if it is a diploid organism, you have a 2C DNA content. If it is a haploid organism, so you should have half, that is 1C. So, some tissues such as the liver occasionally are polyploid in nature, so you have 4C or 8C. So, each species inside the animal, like living kingdom has a particular content of DNA that is constant in all the species. So this is known as the C value. So when it comes to C value paradox, I will just tell that to you in some time. RNA of the chromatin is around 5 to 10%. That's around rRNA, mRNA, and tRNA that is present over here. Now, what are the proteins in the chromatin that are present? Histones, non-histones. Histones are H1, H2A, H3B, H3A, H3 and H4. I mean H1, H2A, H2B, H3, H4. Basic because of their positive, what do you call it, and like, the amino acids that are present. Mostly you have arginine and lysine that are present. You do not have tryptophan, and you have more of amino groups, amino-rich amino acids in fact, and 5 types that are present in eukaryotes. And usually They are making around 60% of the total chromatin protein, and is as equal to that of the DNA that is present. And throughout the history of the evolutionary history, it was seen that H2A, H2B, H3, H4 have not seen much changes during the evolution. So if you take any kind of eukaryotic H2A, H2B, H3, H4, they are like extremely similar, if not the same. So there was a high conservation during the evolutionary history, and they play the most important role in the chromatin organization. And this is rigidly conserved protein H1, in fact, and it is present only once around 200 base pairs of DNA are present, and it is going to help in the coiling of the DNA after, around the histone proteins. It is going to help in binding the DNA. That is the role of H1 histone, in fact, but it is absent in yeast, that is Saccharomyces cerevisiae. Now, when it comes to the protein amount that is present, that is not the not the histones, you have around 20% of it, and 50% of the non-histones are usually structural proteins such as actin, tubulin, myosin. Again, actin, myosin, tubulin, different types are there, like L and B tubulin is present. So what do they do? Help in the condensation and movement of chromosomes during mitosis, meiosis. So they have a structural role as such. And 50% of the non-histone proteins are the enzymes that and different kinds of factors that help in the transcription factors, EF, elongation factor, initiation factor, and regulation factor, etc. They're all again proteins, right? So they are involved in replication, transcription, regulation of the transcription, and they're not going to be very highly conserved, and they're different for different organisms. So function of the chromatin, if you get to see, this is again passing down the genetic information. DNA is the only permanent component of the chromatin, and it is a sole genetic material, right? And so again, mitochondrial DNA is different, and your normal cellular DNA is different. Mitochondrial, like I told you, only maternal inheritance is seen, and it never then the genetic material never leaves the cell. So what is going to happen is heredity of the cell, that is, it is going to be passed on from one cell to the next cell generation is going to it's going to maintain. So when we get to see about the chromatin, types of chromatin, we have seen how it is packaged and what forms chromatin, chromatids, and what is it made up of. DNA, proteins, or and this is how chromosomes are organized. Chromatin, condensation into chromatids. They are the chromosomes. So they are the thread-like, elongated. They are present, suspended throughout in the nucleoplasm. Condensation takes place during the cell replication, and otherwise they help in your basic transcription especially. And the first level of packaging involves the binding of the DNA to histones. So they form the nucleosomes. Once nucleosomes are packed, again, second level of packing of this nucleosomes, they form the 30 nanometers thick chromatin fiber. And this highest level of packaging of the chromatin is present in the chromosome, especially during the metaphase stage. Now, again, two types of chromatin are there, euchromatin, heterochromatin, based on how they stay. And heterochromatin is again of two types, facultative and constitutive chromatin. Constitutive chromatin and the facultative chromatin, and this is basically based on whether there is gene silencing or the chromosome silencing taking place. Non-histones, non-histone proteins, we have seen what kind of proteins that are present basically. The histone proteins are basically your H1, H2, what do you call it? H2A, H3, H4. Non-histone proteins are your the structural proteins basically made up of tubulin, then myosin, tubulin, and different kinds of filaments that are present. And once we are through that, then in the chromatin itself, you have the histones that are present in about equal amount. That is 1:1 ratio. So DNA is the permanent component of the chromosomes and sole genetic material in the eukaryotes at least. Now, when it comes to the C value or the C value paradox, we'll just have a look at it. Now, total amount of DNA that is present in the genome. It's measured in base pairs or picograms. So this term was coined by Houston Swift in 1950, and in genomes, in the chromosomes, it is the chromosomes that is present in the haploid cell. So example of C value is around 6.6 into 10 power 5 base pairs. Housefly is around 8.6 into 10 power 8 base pairs. So it is basically that if the complexity increases, the C value increases from C is simpler to complex forms. And if you see, this means that according to this, the amount of DNA increases from simpler to complex forms. So this is the linear relationship that we see between the genome size and the organism, which is organism complexity. Now, different amount of DNA is present in, if you see morphologically similar organisms are present, but still we have different amount of DNA in the genome. So algae mycoplasma, that is algae and fungi. So you see around 6.6 into 10 power 5 base pairs. Mycoplasma, that is fungi, is around 1.1 into 10 power 6 base pairs. Housefly, fruit fly, Drosophila and housefly, they both belong to fly, that is the insect family. But housefly has around 8.6 into 10 power 8 base pairs. Drosophila has only 1.4 into 10 power 8 base pairs. So there is a difference in the organism itself. So this means that if you see, we will come to that. So this term C value paradox is it was given by the by a scientist C.A. Thomas, and especially this was seen during when the DNA sequences were discovered, and at that particular point of time, genome size, complexity of the organisms, all of it was not taken into was not exactly known. So they came up with this thesis that the DNA content in the organisms of an organism's genome is going to be related to the complexity of that particular organisms. And one thing they noticed that it is different in the higher eukaryotes. So in the higher eukaryotes, there is no relation as such, complexity based on the morphology. So based on the genomic size. So this particular whole program which says that, linear relationship between the genome size and organism complexity is existing is not going to help or is not going to be functioning or seen in the higher organisms or higher eukaryotes. So that is why it is known as a C value paradox. So paradox is basically what it means is that it is a seemingly absurd or a contradictory statement. I'm just telling you the meaning of it. So despite whatever proposition it has made, it is not acceptable. So in other words, we say that simpler the organism that is present, then you have small genome. And if it is a larger organism, we say larger genome. So if at all you know the genome size and you're going to see that if at all it is going to be of a large genomic size, then you're going to know that it is a complex organism. So eukaryotic genome Again, coding DNA, non-coding DNA, introns, exons come into a part later again. So coding DNA gives you the protein synthesis, or like, and the proteins in fact that are going to come. Non-coding DNA present usually a lot of it is non-coding DNA. In fact, 98% is of the DNA is not going to be coded. It's only that 2% which is helpful. Now we know that even non-coding DNA has a role in our like, what do you call it, has a role in fact. So if you see over here, birds, mammals, different kind, like, they have just given, this is the genome size, and this is the C value, and you can just see flowering plants, birds, mammals, and fish. So genome size and the complexity is being given. So salamanders, frog family basically, has 40 times more DNA. But are they more complex than the human beings? Which is actually not true. Drosophila and housefly, they are in the same insecta group, but housefly has a higher value, C value than that of the Drosophila. So this is because of the presence of repetitive DNA. So the sequence of the DNA that is going to repeat more number of times, satellite DNA, will actually or is the cause of the C value paradox that is present. Now, we have something called as the C-value enigma now, which was the term given by Dr. T. Ryan in 2001. So, this tells us about the variation in the amount of non-coding DNA present in the genome of different eukaryotes. And this non-coding DNA amount is going to be different for each species. So this explains the C-value paradox, defines what type of non-coding DNA is present in the eukaryotic genome, that is the haploid set, the genome, and what proportions are present. Now we have something called as the COT curve analysis, which measures the complexity of the DNA or the genome. So it was developed in in the year 1960 by two scientists, Roy Britton and Eric Davidson. And what is it based on? The DNA renaturation. So cut curve analysis can be done whenever you do your RT-PCR. So the rate of renaturation, when you do RT-PCR, you have denaturation, annealing, and again renaturation taking place, right? So in this process, whenever you have the renaturation taking place, this rate of renaturation is going to be directly proportional to the number of times that particular DNA sequence is present in the genome. So if you have the time, then the DNA is denatured. Then again, annealing or reassociation is going to take place in a given DNA sample. So if at all more times same sequence is repeated, that will take less time for renaturation because it knows where it has to go and get attached or get renaturated or gets annealed, like, what do you call it, the formation. So over here you can see heating, re-annealing, cooling. This is like, denaturation. Strands are separated and then annealing, cooling. Double helix renaturation takes place. So large DNA molecules are going to take a longer time to anneal. So what is this COT value now? So the renaturation depends upon the DNA concentration. What is the temperature of the reassociation or renaturation taking place? What are the number of cations that are present? And at the same time, the viscosity or the, like, the thickness of the solute concentration basically. So COT is the DNA concentration you're going to measure. That's going to be moles per liter into renaturation time in the seconds, and you're going to take into consideration the buffer factor. So this buffer factor is basically the effect, of the cations on the speed of renaturation. So again, COT is a concentration of DNA into time basically. So that's what we are looking at. So concentration of DNA, and T is the time taken for renaturation. So low COT value shows that there is more number of repetitive sequences because it takes less time for it to get renaturated again. If there is high COT value, then it shows that it is not of the same sequence, or less number of repetitive sequences are there. So this is what we talk about COT value, that is concentration of the DNA into time. For example, you have 99.7%, which is a single copy bacteria that is present. And if you see in a mouse, you have 60% single copy, 25% around middle repetitive, and 10% highly repetitive sequences that are present. So how are you going to calculate that? Based on the different types of nucleotides that are present, the time to renaturate, say around 300, 400, whatever number of in the seconds. What is the buffer factor? What is the molarity and the viscosity of it, you're going to calculate, and you're going to get the COT value. So DNA concentration into renaturation time into the buffer factor. So how is it going to be? You have a simple COT curve that is going to come up. That gives you, that is, and you can actually plot a graph which shows showing your genome reassociation and the COT curve. So where is it that you're going to apply this COT curve analysis? To just know the number of sequences that are there, when you just do this basic DNA COT curve analysis, you're going to understand what kind of DNA is there. Is there a lot of highly repetitive DNA present? How much amount of it, or how much percent of it is middle repetitive sequences? And how many of the sequences are single curve? All of this with just one particular graph, you can actually get it. So complexity, relative proportion of single copy, and relative repetitive sequences can be known. So for knowing the C value paradox and your COT curve, just go through white and white and Leninger. Principles of Biochemistry. This is one, these are the two books best for biochemistry of any kind of, whatever you want regarding the biochemistry, you'll get it in both these books. Now, let me come to the nucleosomes. Why should they be packed basically? Linear DNA molecule. I might be repeating, but please bear with me. This DNA is condensed with histones, non-histone proteins. So why is it that we need to actually condense the DNA? Because it is around 3 meters long and we are putting it into a cell which is smaller. So we are going to package it in such a manner that it only becomes into the nanometers or angstroms. So when DNA in every cell is 3 meters long, you can just imagine what kind of DNA we have within our cells and how many cells we have. And it is just basic dynamics that you'll have to understand over here. Now, again, different types of packaging: first order, second order, scaffold loop, chromatid, chromosomes. So you have seen this is the DNA double helix, 20 angstroms, beads on a string type of nucleosomes. So this is the first order of packaging into nucleosomes. This is around 110 angstroms. And the second order of packaging is into the solenoid type. This is around 300 angstroms. And this solenoid is present in a loop form again. Each solenoid will form different loops. In this is around 3000 angstroms, or it forms a scaffold loop. And this is again condensed into a chromatid, which is present in the chromosomes. So this is again around 7000 angstroms. And this chromatid is what is made or present in the chromosome, and makes up the different arms of the chromosomes. So when it comes to the structure, like I told you, 8 octamer histone, H2 A, H2B, H3, H4, 4 parts, and DNA. And this is bound by the histone protein. You have the linker DNA over here. So fundamental repetitive units basically. And the formation is again core particle, chromatosome, into nucleosome. And chromosomal proteins, again of two types, histones, non-histones, and they're the most abundant proteins. And they do not have tryptophan. This you will have to remember. You have positive amino acids, that is lysine and arginine, but you do not have aromatic amino acid, which is tryptophan. And highly modified proteins, again of two types, H1, H2A, H2B, H3, and H4. Now each histone is basically, see, it is an amino acid, or like it's a protein. So it has an amino terminal and a carboxy terminal. The amino terminal is going to be hydrophobic in nature. The carboxy terminal is going to be hydrophilic in nature. Now, and it has a central globular structure that forms a central molecule. So together, this H2A, H2B, all of this form the octamer. And what is the component of each histone protein? You get to see it is mostly serine or lysine rich, and H3, H4 arginine rich. Again, bondings is present over there in between them, so that there is the peptide bonds that are present. In fact, they are going to help in a much more compact structure. So, what are the functions of histones? Increase the compact nature of the DNA, and when this is there, it suppresses the genetic activity as such, and structurally make the DNA compact. Non-histone proteins, of course, like I told you, they play an important role in transcription, and they're quite different from the histones. And you have acidic proteins also, and they are going to that, and yeah, that's what they have, the phosphate group in it, which is going to bind to the positively charged histones. And what are the functions of the non-histones? Regulatory, enzymatic. Regulatory because the transcription factors, regulation, like your initiation, elongation, the regulation, all these factors are important, and they play a positive as well as a negative role in gene expression. And when it comes to the enzymes that are present, all enzymes here, polymerases, nucleases, and proteinases, whatever you call, most of these enzymes, they're all the non-histone proteins that are present. Now, the negatively charged phosphate of the DNA and the positively charged lysine or arginine that is present inside the histone is going to form the bond over there. And this is going to make the core electrostatic interaction within the the nucleosome. Other interactions are the hydrogen bonds that are present between oxygen in the phosphate group PO4 minus 3, right, and histones. So you have a hydroxy group that is the it is the hydrogen bonding that is taking place here, and at the same time you have a peptide bond, positive, negative, positive, that is taking place between the phosphate and the positive charge of the in the lysine and the arginine. So, because of this, you can understand that DNA is not going to be within that particular core, octamer, but it is going to be quite widely separated. When it comes to the solenoid model in a nucleosome, so this is all about the nucleolus and the nucleosome that we see. I think we've already covered this, so I'm not going to go much into this. The N terminal is going to direct the DNA to wrap around the histone octamer disc. So the amino terminal is necessary for the formation of the 30 nanometers fiber, and multiple hydrogen bonds are also going to be present, which stabilize the 30 nanometer fiber, DNA fiber in fact. So these hydrogen bonds are going to stabilize the histone octamer present. Now, when it comes to that, you have a phasing of nucleosome, because of which what is happening is, if there is no specificity or if there is a little bit of specificity in the distribution of DNA sequences, then lack of the random distribution is called as nucleosome phasing. So nucleosomes are present along the length of the DNA, and it is not random, but it is in a regular manner. So if you take an base pairs, which is going to be present in the replication region. Promoters are naked, that is, they do not have nucleosomes at all. So this is how it is arranged. So 5' end of the genes are naked. That means that the active genes, whatever is present over there, has to uncoil, and the nucleosome structure is not going to be present at least during that uncoiling phase. So when it comes to modification of the nucleosomes in the active genes, what is happening is you have a acetylation, deacetylation taking place. So lysine will get an amino acetyl can get an acetyl group. So what is happening over here at the amino terminal, you have the electrostatic binding taking place with the phosphate. So because of which this causes acetylation. So in case you notice, this is the amino terminal in lysine, H4. H4 is rich in lysine, right? So over here you have the hat, acetyl hat, and this is a reversible reaction. So whenever there is acetylation of lysine taking place, this binding of phosphate is not taking place. Instead, the amino end has the acetyl group attached. So you get the epsilon N acetyl because it's from the N as N end or the amino end N acetyl lysine being formed. Whenever there is deacetylation taking place, then you have the electrostatic bonding between the DNA and that is with the phosphate group of the DNA and the amino group of the lysine. So this releases acetyl coenzyme A into the medium. So acetylation of conserved lysines is taking place. So this is one example of modification of nucleosomes in the active genes. Let me pause share. I want to share one more, what do you call it, PowerPoint with you. Now we have seen C value, we have seen nuclear structure, nuclear pore complex, we have read about the C value paradox, we have seen nucleosomes. Now we are going to have a look at uh, the giant Lamprush chromosomes or the polytene chromosomes. Now this is basically where we are going to look at see that, please give me one moment. Heterochromatin, euchromatin, centromeres, we have had a look at it. I will have to tell about what do you call it, telomeres also. So yeah, resume share. We have seen heterochromatin, euchromatin. Now I'm going to talk about the centromeres. Condensed region, chromosomes are present basically, replicated during the chromosome, accurate segregation of the chromosome that's been there during your cell division, that is during your mitosis and meiosis. Centromere is going to be darkly stained. Actual location is the attachment of the spindle fibers is the kinetochore composed of both DNA and protein, and then it is known as the sen DNA because sen DNA is centromere DNA can be moved from one chromosome to another. Still, it will help in giving that particular chromosome the ability to segregate. So these sequences will not have any other function. So typically the centromere or DNA is around 120 base pairs DNA has again say, other subdomains CDE1, CDE2, 3. So in this you see this is the Saccharomyces cerevisiae centromere which is around 125 base pairs. So CDE1 has what do you call it it has two conserved sequences CDE1 and CDE3, and you have a CDE2 which is around 78 to 86 base pairs, and it has mostly adenosine, adenine and thymine rich DNA. So the sequences are consensus sequences. They help in, they were derived basically once this centromere was analyzed. Individual yeast chromosomes were analyzed. So usually it is adenine or guanine. PU is adenine or guanine over here. And Y is any base pair can be there. So these two are conserved as such. So you're going to find this particular sequence as such. So you can actually refer the Cooper for this particular information. And when it comes to mutations, in the first two domains, you do not have any kind of effect on the segregation. But in case there is any point mutation in the CD3 domain or subdomain, then the chromosome, centromere is not going to function properly during the chromosome segregation. So we can conclude with this that the binding of the spindle fibers to the centromere in this particular action, CDE3 has an active role. And now when you look at the protein component in the kinetochore, you have three proteins which are CBF3 and this binds to the CDE3 region. It can, but then it cannot bind to a CDE3 region whenever there is a point mutation. So this shows that CBF3 binding is important for DNA or chromosome segregation. So mitotic segregation, I'm not talking about meiosis over here. Please remember mitotic segregation. You have a complex of proteins which is called as CBF3. Binding of CBF3 to the CDE3 region causes mitotic segregation. Further, you see that any kind of mutants of the genes that are going to encode for the CBF3 protein eliminates the ability of the chromosomes to have any segregation during mitosis. Now, when it comes to telomeres, they are the region of DNA at the end of the linear chromatic chromosome. So, what is it necessary for? Telomeres. Replication, stability of the chromosomes. Apparently, it has been from the latest of the news that was released a few days ago, in fact, maybe a month ago. There was a person who spent, say, 3 months plus 3.5 months underwater. And the telomeres shortened, because of which he had age reversal and he became, or he looked as if he was 10 years younger, and that was his cellular DNA age when calculated was 10 years younger to what he was earlier. So if he was in 50s, it looked as if he was a 40-year-old man. That was the cellular age. And astronauts, whoever spent more time in the space, have a lengthening of telomeres. So it plays an important role in the stability of the chromosome, at the same time aging. These are two say features of the telomeres. So Barbara McClintock released that if two chromosomes were broken in the cell, ends were sticky and end of one could attach to the other and vice versa. Now she did not get to see that attachment of the broken end to the end of the unbroken chromosome. So it showed that the end of the chromosomes have unique features. In fact, she had won the Nobel Prize, and she was the one who had suggested about the jumping genes, Barbara McClintock. So telomere sequences are conserved throughout the vertebrates, and they form caps that protect the chromosomes from the nucleases and other influences and prevent the ends of each chromosome from fusing with one another. It is having tandem repeats. You have different kinds of repeats, in fact. So tandem repeats is one, VTNRs are one, and you have your satellite DNA one. So the telomeric DNA has tandem repeat sequences that is present where X, the T by A, X, G by X is 1 and 4, and Y is usually greater than 1. And human telomeres have around the sequence TTAGGG repeated around 500 to 5,000 times. And other bacteria, certain bacteria have the telomeres in the linear genetic material, and they're of two types again. Hairpin telomere, that is, they have a hairpin loop or bend at one end of the DNA strand that is complementary to the other DNA strand. And other type of DNA that is present is the invertron telomere, and this again acts or allows to an overlap between the complementary DNA strands. Now, this is one thing. Now, the last phase, of our topic is basically lamp brush chromosomes or giant chromosomes. So, what is this that we are going to see over here? So, chromosomes only that are decondensed during the interphase. These are special cases of chromosomes that are present. So these giant chromosomes are going to be very long, so and thick. During metaphase, you can see this. That is because of which they are known as giant chromosomes. In both these chromosomes, it is actively synthesizing RNA and it is not condensed properly. It is the least condensed. So example of this is the lamp brush chromosome. It was discovered by Ruckert in 1892. Is seen in the oocytes, egg cells of the vertebrates as well as in some invertebrates. So what is the function? If you see, it produces a lot of RNA and the cytoplasmic and the nuclear volume increases. Now during this, if you see the detailed structure of the lamb brush chromosome was seen or studied during the meiotic division, and in the meiotic division itself you have different phases: pachytene, zygotene, diplotene, and it comes to the diakinesis. So in the diplotene stage of the meiotic division, the structure of the lamb brush chromosome was seen. So large loops of DNA is present, causing this particular DNA to look like that of a lamp brush. And in under the light microscope itself, it can be seen. So this is how the lamb brush chromosome looks. So you have a telomeric loop over here. These are the side loops. And this is the chromatid without any loop. So these have two chromatids, in fact, the lamb brush chromosomes. And the main axis has granules which are chromomeres. They are held together by axial fibers. So you have around many lateral loops that are present, and 1 to 9 loops are present of different sizes, and they are held together by a chiasma. And this is in the form of a paired chromosome, and they form mirror images. And this stage can be present for many months at a time. So this is the central axis, and these are the loops that are present. So when you see the detailed structure of a lamb brush chromosome, this is the paired lamb brush chromosome that is present. It is around 0.5 millimeters. This is the chiasma which is present. And this is the region of the single chromosome having a loop, chromomere. This is the axis through which it is present. We have just enlarged this particular region. And if you enlarge further, you get to see that it is made up of around 10 micrometers extended chromatin, which is in a loop manner, joined by adjacent chromomeres. And this chromomere is formed from highly condensed chromatin. Now, transcription of this lamb brush chromosomes occurs along the whole loop or parts of the loop. And beginning of the meiosis, DNA replication is complete. Homologous pairs are going to be present next to each other, and they are made up of 4 chromatids. So they are distinguished especially because they have a high rate of RNA transcription. So all this RNA transcript is going to be formed on the decondensed loop of DNA. So direction of transcription is also given. This is how the transcription is going to take place. So what is the function of a lamb brush chromosome? Involved in RNA synthesis, protein synthesis. Believed to have one operon, and it has repetitive cistrons, and it has codes for locus codes for the RNA. And Because of this, you're going to get a high repetitive gene sequence also. And where is it going to be? We have already discussed that it is present in oocytes. So it helps in the formation of yolk material in the eggs. Now, we have something called as the polytene chromosomes. So it was discovered by a scientist Balbiani in the 1800s in salivary cells of a particular organism, which is Chironomus, an insect. And it also occurs in the rectal epithelium and the Malpighian tubules. So again, epithelium basically. Malpighian tubules also has epithelial lining only, in fact. And there are many times larger than the normal chromosomes. They reach around 200 micrometers in length. They are going to be visible even in under our compound microscope. In fact, in your BSc, if you see, you have polytene chromosome is one of your spotters. This is because of the chromonemma duplication which do not separate. When there is more separate, replication, but no separation, you can easily or clearly see that because the size is becoming larger. So the polytene chromosomes have 1,000 times more DNA than your normal chromosomes. Uh, Please remember that I am almost done with the class. Another 2 minutes and we'll be done. Because of these chromosomes, basically, they are known as polytene chromosomes. So when it comes to how they are looking at, it is like around 5 long, one short arm, which radiates from a central point, and it is again fused. All the 8 chromosomes, the centromeres of all these polytene chromosomes are fused at the centromeres. So they appear like many arms radiating together. So you have 6 arms, right? 5 long arms and one short arm. This represents or shows the 4 chromosome which is fused and one longest that shows the sex chromosome, that is your XY chromosome. And all of this, you have around 80% of this DNA is present on these bands, 15% between the bands, the interbands. So do we have a picture? Yes, we'll come back to that. So when you see, this is the structure of the polytene chromosomes. They are intensely stained and more condensation, darkly stained DNA bands, less in the interbands. And high degree of packaging is present and they are heterochromatin, so genetically inactive in nature because it is highly condensed. And at the same time, this less packed DNA, whatever is present, it is usually the euchromatin, genetically active. So this is how the what do you call it? The polytene chromosomes are seen. 5,000 bands are present in the 4 chromosomes of salivary glands. Chromomeres are at 90 degrees to each other of the long axis of the chromosome. So DNA content is high, so they also absorb UV light, and then these bands, whatever you see, usually are corresponding to the genes that are present. Where are they present? These polytene chromosomes, salivary glands of and other tissues, the Malpighian tubules and your other epithelial cells in the flies. And when is it seen? Usually during interphase. And how is it arranged? In alternating bands and interbands. So this is the larva of the chironomid. And in this you have the salivary glands that are present. And this salivary glands DNA is in the form of a polytene chromosome. This is the chromomere that is attached. Now, so in detail, you see this is the chromosome, and when this is what do you call it, seen, it is the highly condensed form, right? So you have chromomere, chromocenter 1, and then you have this is the X chromosome having the homologous chromosome separated during normal mitosis itself. If you see this, you have the right arm of the chromosome 2, left arm of the chromosome 2, around 20 micrometers if you just measure the length. And this is the chromomere, chromocenter, I mean, and they're the separated homologous chromosomes. And then you have the chromosome 4, right arm of chromosome 3, left arm of chromosome 3 attached at the center, chromocenter. Now, what is its function? Does it have a function as such? During the larval development of Drosophila, a lot of it are going to be in a temporary, phase basically in the polyp chromosomes. You can see them as puffs. These are again decondensed, expanded, and form the active chromosomal regions. And these will get into transcription or and so on. So location of this particular polytene puff shows the different stages of larval development. So yes, we can understand or know the phase of the larva, seeing the cell, what do you call it, polytene puffs. And when you radioactively label this RNA, then you know what kind of gene is being transcribed, and you can know what kind of gene is going to be coding for what protein. So this is the chromosome puff, and these are also known as the Balbiani rings, named after the scientist, in fact. So these puffs are associated with metabolic activities helping RNA synthesis. So labeled RNA, this is how it is going to be for functional stages in the polytene chromosomes, if you see. So increase the volume of the cell's nuclei. Last two slides and I'm done with this class. So they increase the cell's nuclear volume. So it causes cell expansion, and multiple copies of genes permit high gene expression, in fact. And in your fruit fly, that is Drosophila melanogaster, these undergo reduplication. So what is happening is large amounts of glue is being produced before the pupation stage or pupal stage. Now tandem duplication, that is duplication of these polytene bands that is present near the centromere of the X chromosome, results in the bar phenotype and having kidney-shaped eye. So it is this tandem duplication of these polytene bands will cause the bar phenotype in the Drosophila to come. So Drosophila, development, you have different kinds of stages. So in that, the bar kind of phenotype that is being expressed is because of duplication of the polytene bands. So these are the referral, books that you can actually see. You have different kinds of journals. You can actually browse it inside the net also. We are done with this class now. Yeah. So that's about it for today's class. We'll meet next week. And whatever, PowerPoints I have shown you as of now, I will share it with Kishore sir. You can just have a look at it. As it is, actually, this lesson is being recorded. So you can go through all the PowerPoints that are there, whatever I have shown you once you see the video. Otherwise, also, whatever I am taking the matter is actually from the net only. So you can just have a look at it. How to join the WhatsApp group? Please ask Kishore sir if you have Kishore sir's number. So you can just join the WhatsApp group over there. And I will share this particular PowerPoints, like I told you, whatever presentation it is there with Kishore sir. Unit 2, 3, 4 I did. 5, 6 I will do it like within this week. All right. Yeah. Thank you so much. Yeah. Thank you.
Thank you, ma'am. Thank you.
Pleasure.